Thanks, Dr. Weimer. So it's really my privilege to give a short biography and introduction for Dr. Kuenzel. He's a very, very important person in my life. I literally would not be here if it wasn't for him. So Dr. Kuenzel was raised in New Jersey, but ultimately he wanted to go to West Point, but that didn't work out for numerous reasons, and which turned out to be for the better. He ended up doing his bachelor's at Bucknell University in Pennsylvania, and he began taking biology classes and met who eventually became his master's advisor, Dr. Carl Helms. And this is a picture here of Dr. Helms. And Dr. Helms was actually a student of Ernst Mayr, who those of us who are ornithologists, birders, are very much aware of. He's a very famous ornithologist from Germany. And so I'm almost getting goosebumps just being that close to Wayne. He did his, Dr. Helms was then offered a position at the University of Georgia, and Wayne followed him there to do his PhD, where he investigated the role of the hypothalamus in food intake, fat deposition, and migratory behavior in, I believe, white-crowned sparrows. I believe, yeah. And after his PhD, when he was at Bucknell, he was a part of the military program for classes, so he owed the Army 2 years. So he then went to the Fort Sam Houston Army base in Texas, where he was a captain, and he instructed field techs and medics for doing war surgeries and battle repairs. And this was in the Vietnam era. Fortunately, he stayed in the States. But as you can see, he really, really embraced the lifestyle of Texas at the time. After his stint with the Army, he decided he really missed research and wanted to go back. And he went to Cornell University, where he studied under Dr. Arie van Tienhoven. And for those of us who are neuroscientists, that name just gives me goosebumps. Dr. van Tienhoven is the individual who pretty much defined the neuroendocrine regulation of puberty and defined what puberty is physiologically. So absolutely fantastic opportunity for Dr. Kuenzel to learn about the neuroendocrine regulation of reproduction under van Tienhoven. After his postdoc, he joined the faculty of the University of Maryland, and he was there for 26 years, where he ultimately became a full professor. And he met many influential people, like one of his students, Nimish Vyas, and who also studied migratory behavior in white-crowned sparrows, and of course his wife, Kim, he met while at University of Maryland. And eventually he met this really annoying kid who, I don't know about all of you, but I wouldn't invite that kid into my lab. But he did anyway, and well, I guess it kind of turned out for the better, for me anyway. But after 26 years at the University of Maryland, an opportunity arose here at University of Arkansas, so he moved here, and this is actually a picture of him moving his family. You can see his wife here, oops. And his 3 children. And I'm not sure where Rain is in the picture, but I'm wondering if that's his foot sticking out of the grill of the car. But he has been here for 24 years, I believe, and has continued his work studying the neuroendocrine regulation of puberty and reproduction, as well as the stress axis. And his accomplishments over the last few decades are just simply too many to count. But I want to highlight 2 of them. First, in 2012, he was inducted as a fellow of the Poultry Science Association. And this picture on top is where a bunch of his students attended, including myself, Dr. Mary Beck, Dr. Farrah Madison, who's now faculty at University of Wisconsin, and as well as his wife and 2 daughters. And so that was really wonderful. And recently, just last month, he was inducted as a fellow of the American Association for the Advancement of Science, which was truly a wonderful accomplishment, and great kudos to Dr. Kuenzel. And I, again, had the privilege of being there with him for his support. But this morning, as Dr. Weimer already indicated, he's going to be talking about the importance of the septal brain area of birds for optimal neuroendocrine regulation of reproduction and stress. Now, Dr. Kuenzel wasn't sure if he was going to be able to make it this morning, so we have actually pre-recorded his talk. So everything is all set that we will just show this video, and then he'll be available to answer questions. Can you help me?
My name is Wayne Kuenzel. I work in the Poultry Science Department, and I study recently the reproductive system and stress. So the HPG axis in mammals involves the H for the hypothalamus, the P for the anterior pituitary, and the G for the gonadal axis, involving the specific compound GnRH, gonadotropin releasing hormone neurons. And so apparently, according to physiologists, it's the HPG axis that's responsible for regulating reproduction in mammals. And the avian community likewise has agreed to it, that we, that is, in the avian system, has a similar one to the HPG axis. However, there's another structure involved. What's different about the avian HPG system is that there's another brain area involved. It's called the septum. And the septum is just dorsal to the hypothalamus, and it has a critical region. In fact, there are two cell areas, the septal medial and the septal lateral nucleus, that is loaded with gonadotropin releasing hormone neurons. And that's what makes it a little bit different from the GnRH system, in mammals. The septum ought to be included, and so the abbreviation should be the SHPG axis in birds. And the S is for the septal system, that's activated first, followed by the hypothalamus, anterior pituitary, and final activation of the gonadal system, producing GnRH in that system. The septum is another brain region that's very much involved in the two axes that we're talking about, the one that drives the reproductive system and the one that drives stress. And because of that, I believe that there ought to be a change to the abbreviations that describe each of these two processes, and that's part of this talk. So that today I'm hoping that you will accept the idea that for the reproductive axis, the abbreviation should be, instead of just the HPG axis, it should be the SHPG axis because of the role of the septum. And also for the stress response, where we have the HPA axis in mammals, it's the SHPA axis in birds. And so please keep that in mind, that in the avian system, the role of this different brain region, the septum, is important and should be included in the abbreviations of those two systems. Well, one thing that's interesting is that in both um, mammals and in both birds, the system is in a sense quite similar in that the GnRH neurons did not originate in the brain. They originate in the embryonic olfactory placode. This is located just ventral and posterior to the olfactory bulbs. This is where they then enter the brain from that olfactory placode, and from that point on, they move posteriorly, passing the olfactory tubercle, and from the olfactory tubercle finally to the septal region, which is close to the middle of the brain of birds. And so it's hard to believe, but these neurons, once they enter right after the olfactory bulb, they then have to move quite a distance and go from the olfactory bulb way in the front of the brain and go all the way back to the hypothalamus. And that's where they end up in the septal region. And so this is quite a migration, but both in mammals and birds, these neurons do not originate in the brain. And a lot of people are not aware of that. And also, there are chances that all of these neurons will not make it to that septal and hypothalamic region for the production of GnRH, in birds. It seems to be the difference is that in mammals, it's strictly the hypothalamus involves all of the GnRH neurons. However, in the avian system, the septal region, in addition to the hypothalamus, has that GnRH system. The reason there is a deep brain, or talked about deep brain photoreceptors, is that it really is, these receptors are very deep within the brain. That is, they're not on the surface, but you have to go all the way down into the base of the hypothalamus and also the bottom of the septal region in order to activate these neurons. When you see that term deep brain, don't let that bother you. It just means that the components of the photoneuroendocrine system are not at the top of the brain, the dorsal region. They're all the way at the very, very bottom in an area that finally ends up being the median eminence, which is the output region of the ventral brain region. And that means that when you increase the photoperiod, that light has to penetrate through skin, bone, all of the brain, and finally activate these neurons way back down in the diencephalon or hypothalamus. And that would be the best way to incorporate that. And yet it works. And the light is able to penetrate through skin, through bone, through the entire brain, all the way down to the base of the brain to activate it on long day photoperiods in the spring. And that's when this system is activated in birds. So the photoneuroendocrine system involves activating the reproductive system. And anytime you think about reproduction, then you should think about GnRH, gonadotropin releasing hormone neurons. These are the ones that are the neurohormones that drive the reproductive system, not only in mammals, but also in birds. And so what we have found in both groups of vertebrates is that they're very sensitive to photoperiod. And particularly in birds, if you increase the photoperiod, you cause the development and increase in the number of gonadotropin releasing hormone neurons. And so therefore, as we can see in the springtime, with an increase in GnRH neurons, that system is activated and the gonads begin to develop rather quickly. So that's what makes it quite an interesting system to study in detail. When the photoperiod increases in the springtime, what that also means is it activates the photoneuroendocrine system, and that involves GnRH neurons, but also these so-called receptors and other components that are at the very base of the diencephalon, which is the term for incorporating the hypothalamus, where the GnRH neurons end up. So please keep that in mind, that light has to penetrate through the whole brain all the way down to its bottom in order to activate it during the springtime or long photoperiod times of the year. As you might know, that it takes a lot of energy in order to develop the gonadal system and the thyroid system. So just as there's a hypothalamo-pituitary gonadal system, there's a hypothalamo-pituitary thyroid system that provides the energy to drive this system. They provide the ATPs necessary so that they're ready to go, and the gonadal system is activated because of that supply of energy. The other one that we're discussing is the stress pathway. The stress pathway can be any time of environmental change that impacts a large group of birds or mammals, whichever you're talking about, and that takes energy as well. And again, the thyroid axis provides the energy for the stress pathway when that's activated. So here we see the thyroid really paying attention and critical for the success of the reproductive axis, and it provides the energy for the proper response to stress due to an environmental change. That environmental change could be time of year, but it also could be within the system. You know, a whole new group of birds coming and joining a particular group will upset the organization, and yet they're the same species. You have to learn to adjust when these kind of environmental impacts occur in both birds and mammals. The mammalian system for the HPA axis, so that would be the hypothalamo-pituitary adrenal axis, involves the production of cortisol in mammals, corticosterone in birds. So it's not that different from the HPG. That is, within the hypothalamus, they have the neuropeptides responsible for activating that system. The peptides would include two of them. One would be the corticotropin releasing hormone neurons, as well as arginine vasotocin neurons. And so these two are required in order to activate the HPA axis. And in both the mammalian system and in the avian system, they both have CRH, which is the main one, In the mammalian one, it's a vasopressin, but in the avian one, it's arginine vasotosin. And the primary one is the CRH, because that's the one that's activated first, at least in mammals, to drive that system. But birds are different because there is a septal brain region. So in the septum then is a small nucleus called the nucleus of the hippocampal commissure, and it's loaded with CRH neurons. That drives that axis, the stress axis in birds. So that should be included. So instead of just having an HPA axis, it should be an SHPA axis in avian species. CRH is activated first in the stress pathway and begins that whole process of trying to get the energy and systems going. So CRH is produced first, but then after that, AVT needs to be produced to maintain that stress pathway. In mammals, it's the HPA axis we're talking about, but note that in birds, it's an SHPA axis, the septal region first, and after that is the hypothalamus, then anterior pituitary, and the adrenal axis. And what the septal region has is the nucleus of the hippocampal commissure. This very small nucleus is just above the hypothalamus. It's a small nucleus, but it's completely filled with, in the dorsal region, with corticotropin releasing hormone neurons. And that's critical here to drive the system. It always has a peak in CRH first with that system. Then the hypothalamus comes along and it takes longer, and there's a second peak of CRH, but that comes from mainly the hypothalamus, not from the nucleus of the hippocampal commissure, and then finally arginine vasotosin from the hypothalamus. So you can see it's sort of a three-step. So you've got the septal region, but you have also the hypothalamus having the same two CRH neuron groups that start to activate the system. However, to maintain it, particularly for chronic stress, you need that arginine vasotosin, and that's the third peak that keeps the production of all of these. Will increase adrenocorticotropic hormone from the anterior pituitary. And that hormone here will then drive the adrenal gland at the very bottom for the production of corticosterone. And it's corticosterone which is the stress hormone of birds, corticosterone. You need to know that CRH appears to be the first neuropeptide that's activated in the system. And then the point is, because stress can be acute, but stress can also be chronic. And when it becomes chronic, this is where the vasopressin system in mammals and the vasotosin, arginine vasotosin system in birds is critical. It continues that process with the production of arginine vasotosin. The arginine vasotosin, the second peptide, is the one that has to maintain that system long term, and that continues that important stress response in order to handle whatever has occurred in that population whether it's an increase in their number or something else occurred in their system that drives this response. That's when arginine vasotocin comes in, and that will continue to increase in abundance in order to make sure that there's sufficient corticosterone produced, as well as the anterior pituitary production of ACTH to keep this whole system going. In this system, it's always CRH that's activated first, and it's only during chronic or continued stress where the importance of AVT is critical to maintain that system throughout that stress period. Yes, there are, and interestingly, and the male response is greater than the female response in the production of the peptides as well as ACTH and corticosterone when the system is stressed, at least in birds that have been examined. Yes, there is, because just as the gonadal system needs energy, the stress pathway needs energy for maintenance. And so the thyroid will need to be incorporated to provide the energy to drive the particularly long-term stress system. It takes energy, ATPs, to keep that going. And that thyroid is critical for providing that energy. So keep in mind, when you think of the osmotic system, you're really talking about the posterior pituitary getting involved in order to regulate mineral balance within the body. This means that the body is trying to balance some of the minerals such as sodium, chloride, potassium, magnesium, and in that case, the posterior pituitary is involved. So that's truly different because the osmotic system then involves not the anterior pituitary but the posterior pituitary. And so that osmotic system then can be important, and it is important, but it's a little bit different because of that structure involving the posterior pituitary gland and its production. Mobilization, though, is like the other stressors. It does not involve the posterior pituitary. So, for example, the two stressors we tried was food deprivation. You take the food away from a bird, and gradually you can see that without food, their stress area, if you're looking at stress, starts to build up and get higher and higher. With immobilization, it's quick. So you can see that when you immobilize an animal, you get the response quickly. And so those are two very, very different stressors, but obviously both will activate, but it's the feed deprivation that's long-term. And so that's a difficult one for the system to handle long-term. But there's no choice but to try to keep that system active in order to produce the not only because they're stressed, corticosterone, but that means that thyroid axis has to be activated. And remember, they're not eating and they're going to be running out of energy in order to provide the necessary ATPs to drive that system. I think you have to look at those two systems as antagonistic. So the HPG axis is an axis that's trying to develop the gonadal system, whereas the other one involving stress is something that is activated and energy is required. So the point is they're antagonistic in that sense. The other thing that's interesting about them is that when we go back into the septum, you'll notice that the HPG neurons are in the septal region, but they're in a different, they're more scattered, but they are in two nuclei within the septal region with the CRH, that is the hypothalamo-pituitary-adrenal axis. They're only in that tiny area called the nucleus of the hippocampal commissure. That's where the CRH neurons. And so again, they're antagonistic. So you can see they're occupying the same region, but only that little nucleus drives the one system for stress, whereas the larger two areas, the SM and the SL, have the gonadotropic hormones that drive gonadal development. Reproduction. Today I'm hoping that you will accept the idea that for the reproductive axis, the abbreviation should be, instead of just the HPG axis, it should be the SHPG axis because of the role of the septum. And also for the stress response, where we have the HPA axis in mammals, it's the SHPA axis in birds. And so please keep that in mind, that in the avian system, the role of this different brain region, the septum, is important and should be included in the abbreviations of those two systems. So again, that would be my final comment that I would like to make is don't forget the septum. It's a large area. It's directly above the hypothalamus. That's where we find the critical peptides for the stress pathway, and importantly, the critical peptides for the reproductive pathway, not the hypothalamus. They're both located in the septal region. What I'd like to do at this point is to ask for any questions about this talk involving two, and there's more than just two, systems that interact with the anterior pituitary, but also the posterior pituitary is activated as well in trying to drive these systems when you have the basic course in physiology. Normally in the mammalian system, the septal region just isn't talked about, but certainly in birds, please keep in mind the septum. It interacts very well with the hypothalamus, and the two really are important in driving neuroendocrine systems. Anytime you see neuroendocrine, we're talking about not just the hypothalamus, but the anterior pituitary and possibly the posterior pituitary, or both of them involved in the system. So that was part of what I was trying to do to clarify these neuroendocrine systems in birds. So if there's any questions, I'd be happy to answer them at this point.
Does anyone in the audience have any questions?
Thank you, Wayne. That was a great talk. I'm curious, with the GnRH neurons, there seems to be a much larger distribution in birds compared to mammals across the septal and hypothalamus, and even I think some in the thalamus. Does that suggest that there are other roles for GnRH than just the SHPG axis?
Well, there's more than one type of GnRH molecule in birds. There's some of them, they call it the GnRH1 or the GnRH2. The one is the one that activates the reproductive system, but there's a GnRH2 that goes beyond the septum, if you recall those slides, and goes into the midbrain of the bird. So the point is, what's the GnRH2 doing? So they're probably involved either more on energy perhaps or some other issues. So keep in mind that, and then in the mammalians, I think they have 3 different types of GnRH molecules. So these are interesting variations and something to keep in mind regarding that it's a small neuropeptide, but extremely important and diverse. So it's not just reproduction, but there are more than one GnRH type that's involved in birds as well as in mammals.
Dr. Kuenzel,
Yes.
I have a question. Someone else? Dr. Kuenzel? Over here. Okay. Thanks for a fascinating presentation. Being a person whose my area of research and interest is different than this, I was really interested in the discussions you were having there about the spring and the changes in light in the spring and the impact on the reproductive system beginning in the spring. So my question to you is around.
Can you please, I'm having trouble hearing. Can you please repeat that?
Okay, so you were talking in your presentation about the impacts on the reproduction system that change during the spring, during the springtime.
Yes.
Which to me elicits a question around the difference in the penetration to the deep brain. What is the impact of the length of light in the spring versus the intensity of the light? How much impact do each of those 2 factors have, the length of light duration versus the intensity of the light, in making those impacts in the inner part of the brain?
Yes. Well, what's amazing is that the way the brains are organized, it can penetrate regardless of the time of year. But what's important, of course, in the spring is the length of time. So as photoperiod increases, you have more hours of that GnRH, for example, being activated in the spring versus the fall. And that's what also drives the system. So keep that in mind. But it's remarkable to me that light can get through, the feathers, the skin, the bone, the entire brain, and both of these systems are at the bottom. So it has to go through the entire brain in order to activate these neurons, even in the septal region. And yet that light has the ability to go through these structures regardless of the time of year and activate the system. So it's something else to keep in mind, that it's the way it's designed. They penetrate very, very efficiently and can activate at any time of the year.
Thank you, Wayne. If there's other questions, let me know, but I have one. So that's very, very interesting that the light is perceived at the bottom of the brain in avians, and kind of coming off of Bill's question is, I assume there's a certain intensity of light, a minimum intensity that needs to be perceived, right, to travel all the way through. Is that correct?
Well, there probably needs to be perhaps a limiting intensity, but keep in mind that it doesn't matter which time of year it is, it penetrates down, and our systems are activated 12 months of the year, and in other animals and birds, it's the same. There is that remarkable ability of that sunlight to get all the way down to the bottom, and perhaps that's why they were positioned there in the first place, in order to not outcompete other areas of the brain that obviously have to operate it concurrently. If I'm answering your question.
Yes, you did. Do we have more questions? All right.
Good morning, Dr. Kuenzel.
Good morning.
I have a more pragmatic question for you. I thought your comments on the stress response to feed withdrawal and immobilization to be interesting, because we do both of those things prior to live haul. Is there anything you think we can do differently to modulate the stress response to those two events? Taking birds off feed and then loading them into a truck.
Okay, and repeat the question at the very end, please.
Is there anything you think we can do differently in live production to ameliorate the effects of taking birds off feed and then immobilizing them, if you would, in a trailer? I realize that's a very stressful thing we do. What can we do differently to modulate that?
You mean in terms, within the processing plant, how is there a way to better?
I'm thinking during live haul. So the actual event of taking them off feed, putting them on a truck.
Yes.
Very stressful. I realize that. Can you think of anything that we can do differently that might ameliorate the stress response?
That would be a difficult question to answer, because the nice thing about the current system is it's fast. And what you want to do is get those birds, off the farm into the truck as quickly as you can. And I don't think it would take some big sophistication to, I think, improve what the current system is. It's speed that you really want, and get those birds, off the farm and into the truck as quickly as possible. And the same thing, off the truck and into the processing plant as quickly as possible. I think should always be the priority in handling chickens practically.
Thank you, Wayne. And turkeys, Brian. Do we have any other questions? All right, I have one. So it is very interesting, the emphasis that you placed on the posterior pituitary regulating the osmotic system and the anterior for the stress system. How does the osmotic system interact with the stress systems? Are there certain minerals that activate enzymes for the stress response, or can you just speak a little bit on the interaction between the two, or feedback mechanisms?
Interaction between posterior and anterior pituitary?
Yes, sir.
Right. It's a good question. I think the one thing is that I'm sure in some cases, probably both are activated at the same time. But clearly, the one way to keep in mind, and this is the way I try to keep it straight, is generally if there's anything to do with mineral changes, this is where the posterior pituitary will dominate and be important. On the other hand, if it's having to do with stress of, for example, immobilization versus feed deprivation. These are two very different stresses. Immobilization is fast. You know, you put something around the birds so that they can't stand up or fly. That's quick. If you do feed deprivation, it takes minutes, hours. And so if you look at a time for one, for example, like feed deprivation, that takes a long time for it to really just build up and get more and more stressful versus immobilization, which is immediate, the sequence is the same. It's always the nucleus of the hippocampal come ashore that shoots that first rise in CRH. And then it switches to the hypothalamus. And then you have the CRH and the AVT. But even though they're very, very different, the sequence is the same. And I think that's what makes it interesting, that the stress won't matter. Just keep in mind that the sequence of activity of the neuroendocrine system will be the same sequence. And I think that's what keeps it going. And I guess that's all I can say about it.
All right, well, let's thank Dr. Kuenzel.
All right. Thank you very much.
Thank you.